Hi, welcome to the Bioinformatics Chat. Today I'm talking to Geoffrey Schiebinger.
Hi, Roman.
Hi, Geoffrey.
Yeah, thanks for having me here. It's an honor.
Yeah, great to have you. So you recently, with your colleagues, released a preprint. It is titled Reconstruction of Developmental Landscapes by Optimal Transport Analysis of Single-Cell Gene Expression, sheds light on cellular reprogramming.
That's right.
Yeah, this is what I'd love to discuss with you. But before we get to that, can you talk a little bit about your background?
Great. Yeah, I'd be happy to. So I come from a PhD program in theoretical statistics, and this is my kind of first experience in biology. So it's been a kind of trial by fire. It's very intense experience. Very different from what I did during my PhD. So my background is more in very theoretical statistics. I chose statistics to study for my PhD because, I was always a scientist at heart, but I never really knew what type of science I wanted to do. Like, for example, in my undergraduate experience, I worked a little bit analyzing the light from the cosmic microwave background radiation, and also tried to develop an optimization algorithm to track a diffusing protein with a laser beam. But I also, in 2006 and 2007, had some experience pipetting cells by hand and doing single-cell gene expression analysis kind of before it was cool. But now, because of that experience early on, analyzing single cells, after I heard about these new measurement technologies with droplets and 10x, the scale was just so much bigger that it kind of brought me back into this specific type of science, and I'm really happy with how it's gone so far. Okay,
so you came to the Broad Institute, and is this your postdoc?
Yes, that's right. I'm a postdoc at the Broad Institute. I'm in Aviv Regev's lab, and I also work closely with Eric Lander, and I'm also co-advised by Philippe Rigolet in the math department of MIT.
So I'm curious, does Eric does he continue doing actual science, because he's the director, right?
Yeah.
He's probably very busy with the business side of things.
Well, actually, a lot of the math here was worked out on the whiteboard with Eric, and we'd meet, in the evenings or Sunday night, go to his house. He's very involved. He called me on the phone at one point with some idea, and that eventually is how the idea to use optimal transport came out of it, was through intense conversations with Eric.
Wow, that's crazy.
Yeah.
So, can you describe the general sort of problem, the context that you're working on?
Totally, yeah. So the main idea is that you have a developing population of cells, and we model this with a probability distribution that's changing over time, like a stochastic process. And the main idea is that if you take a random cell from an embryo at some specific time, or say a population of reprogramming cells, then its gene expression is random, but it's random in a different way at different points in time. And experimentally, we can measure groups of cells, we can measure populations of cells with single-cell RNA sequencing at different time points. And the goal is to figure out how this distribution of cells is evolving over time. Now, the key problem is that when you measure a single cell, when you measure its expression profile, you actually have to kill the cell. So you can't physically measure all of the genes at once in a single cell over time. Now, of course, you could maybe measure fluorescently a few genes over time in a single cell, but with single-cell RNA sequencing in droplets, where you can do hundreds of thousands of cells with very high throughput and measure all of the genes, this actually kills the cell. So then mathematically, what we have are a set of samples from this developmental process at one point in time and at another point in time. And so kind of for any point in time, we can sample from this distribution. And so at one specific point in time, you could estimate what subpopulations of cells there are, by clustering, or you could maybe, try to get an estimate for the density of this distribution somehow. But the key mathematical problem for us was, how can we couple these distributions of cells at two different time points in a meaningful way? So this notion of coupling is kind of an idea from probability theory that if you have two random variables, then what's their dependence? So people are familiar with the idea of kind of correlation, right? Like, the gene expression profile of a cell at one point in time is probably correlated somehow with its expression, 1 second later, right? So these are not totally independent random variables. So biologically, the cell has some time scale on which its expression is coherent somehow. And so for us, we were trying to figure out the best way to couple the cells at one point in time with the distribution on expression at the next point in time. And this would tell you, like, if you knew this true coupling, it would tell you, given that a cell has some expression profile at one point in time, what are the likely fates that this cell could take? Where might this cell go in gene expression space from one point in time to the next point in time? And the mathematical tool that we're using for this is called optimal transport. So optimal transport is a way to specify a coupling between two probability distributions that minimizes the total cost of the coupling. And the cost is specified in terms of a distance that each cell would have to move.
And we should clarify that cells move not spatially, but in the gene expression space.
That's a very good point, yes.
So you said coupling is a generalization of this notion of like correlation or
right.
interdependence, right? And another obvious generalization of correlation or interdependence is the joint distribution. But I have a sense that coupling has more information in it than joint distribution. But can you explain the relationship between these two?
Yeah, very good point. Yeah, so coupling is precisely the joint distribution. I should have just said it that way in the first place. But for us, we actually modify this notion, this fundamental notion, a little bit, because we're not actually just dealing with probability distributions, which is what optimal transport classically deals with. We're actually, now this is a technical detail, but we're dealing with just distributions, which could have mass different from 1. Okay? And for us, this notion of a distribution just captures the probability distribution over gene expression space where cells could be, together with the total number of cells that we would have at any given point in time.
And so when you say this generalized distribution, which is not probability distribution,
right?
So its total mass is not 1,
right?
And another, I guess, name for that is measure. So is there a difference between these distributions and measures?
No, it is a measure. That's exactly right.
Okay, it's just measure seems to be like a more common term. So was there a reason why you went with distribution? I found a bit confusing juggling between distributions as probability distributions and distributions that are not.
No, I mean, these are just kind of semantics, right? So yeah, mathematically it's a measure. A random variable has a distribution. It also has a cumulative distribution. But for us, there was also the mass. I just use distribution because it seems like a more friendly word. Measure has all this kind of scary technical stuff associated with it, but there's no real deep measurability issues that we run into in our stuff.
Okay. And so the reason why you don't just restrict yourself to probability distributions is, I guess, because of cell proliferation, because you have to account for cells dividing. Is that correct?
Yeah, that's exactly right. So we modify the notion of a coupling, to include growth. Now, just to be completely fair, people have done this before. So Linek-Shiza has a wonderful paper on unbalanced transport. So he, extended optimal transport to the case where the measures don't have to necessarily have total mass 1. They don't have to be probability measures. And for us, what we did was we included this also deterministic notion of growth as well in the constraints. And this modifies the notion of the coupling as producing mass locally and also transporting it. So this is a nice way to think that, given that a cell is at a position in gene expression space, it will have a certain number of descendants, and those descendants will be distributed in a certain way. And so the coupling for us is capturing both of those notions.
And I guess this growth function models not just cell proliferation, but also cell death. Is that something you have to deal with?
Yeah, exactly. We have to deal with cell death. So the way that we think about this is that birth and death together both affect how many descendants a cell will have at the next point in time. And based on, gene signatures for proliferation or apoptosis, you can define some, estimate for how many descendants you think a cell will have. And abstractly, this is this is one of the inputs to our algorithm, is an estimate for how many descendants a cell will have at some arbitrary time in the future.
And when you model the cell proliferation and cell death, you don't try to infer it from the data, but instead you postulate this growth function that tells you the rate at which cells are growing depending on their gene expression profile. So what was the rationale behind this? Why not try and infer these events from the data itself?
So the data that we actually have, that's a good point. So we should base these on data, and the version of the paper that's released, has some estimates for growth and proliferation that we basically just based on kind of biological knowledge, just kind of rough back of the envelope calculations of doubling rates and stuff like that. And you could certainly do something more sophisticated. And, in our, we're actually kind of pushing on that a little bit. But what's not identifiable from the data is kind of the overall rate of growth of all of the cells, because all that we measure at any point in time is the relative abundances of the subpopulations, because we could sample a certain number of cells from this distribution at one point in time, but all we really see are kind of the numbers of samples that land in different regions. We don't see kind of the total, but you could estimate that by looking at the number of cells in the plate, or something like that.
That's a good point. I didn't realize that. So you control how many cells are isolated and sequenced. It's not a biological quantity, so you cannot just infer the abundance of cells from just their count.
Right, and in principle, there could be some biases in terms of which subpopulations are recovered, But we're kind of assuming that we're sampling from some population, and then, based on how many cells you collect at some time point, you can control how many samples you get at that time point.
Right, but your growth function is local, right? It's not that you say, okay, my total population of cells doubled in this period, but you actually try to say, like, depending on where in the gene expression space the cell resides, right? You determine this custom growth rate.
That's right.
So wouldn't that be identifiable if you just said the overall growth rate? Couldn't you determine from the data what are the growth rates in each patch in this gene expression space?
It's a little bit confounded by the fact that the cells are also moving in gene expression space at the same time. So I think in order to explain this, it's going to be helpful if I explain a little bit more about what optimal transport is doing, and also how the growth enters into the constraints in optimal transport when we are trying to infer the coupling from one time point to the next.
Yeah, let's do that. Yeah.
Okay. So imagine that we have a group of cells at one time point and a group of cells at the next time point. Okay. So we view these as distributions on gene expression space, and we're inferring their transitions using optimal transport. So optimal transport has it's a classical subject in mathematical analysis that goes back hundreds of years all the way to the work of Gaspard Monge in the 1780s, who was using it to design efficient plans to build fortifications and move dirt as efficiently as possible. So for him, the total cost of moving a pile of dirt from some distribution P to another distribution Q was specified by a transport function. So he actually wanted a deterministic transport function that says that you take a particle of dirt at position X and you move it to some other position Y. So you want to minimize over all these transport plans, and then Leonid Kantorovich generalized this framework in his study of linear programming in the 1940s by introducing a relaxed version of this problem where you can specify a non-deterministic transport plan, which is more like a non-deterministic coupling of random variables where, given that a random variable X is specified, the distribution of Y is not, it could have some entropy in it. This is kind of the framework that we're using. We're using kind of a modern formulation of this problem. We use this to compute the coupling of cells at one time point to the next, okay, in order to minimize the total transport cost. So the transport cost is it's defined in terms of some cost function. If it costs, for example, if the cost is in moving dirt is defined by the distance that you have to move it times the mass that you have to move. That's the total amount of work, I guess.
I guess I don't completely understand the difference between the deterministic and non-deterministic transport plan. So when we work with probability measures is just a different kind of measure, right? So what makes it non-deterministic?
Okay, so imagine you wanted to transport a Gaussian centered at 0 to a Gaussian centered at 1, okay? Mm-hmm. Here's two ways to do it, okay? One is you pick up a point X and you move it to X plus 1. And you do this for all X. Each X is sent deterministically to one specific point. Okay? Mm-hmm. Now, here's another way to do it. You could pick up this point X and then you could randomly just pick a point Y distributed according to a normal at mean 1. And if you did this for each of a bunch of X's, then the distribution of where they were all mapped to would eventually look like a normal with mean 1.
So mathematically, what does that correspond to? Because when we talk about deterministic transport plan, we have just this joint measure, right? Coupling. And then when we move to non-deterministic, is that a different mathematical object?
No, so they're both couplings. But the difference is just if you fix X and look at the conditional distribution of Y given X, if that conditional distribution is deterministic, then that would be a deterministic transport of X to a specific point Y. With probability 1, right, you would transport X to a specific point Y equals X plus 1 in the normal example.
Okay, got it.
Okay, so I've described the concept of optimal transport and its historical background as kind of Earth mover's distance. Okay, so given some cost function, which specifies the cost of moving a unit mass of dirt from position X to position Y, you can search for a transport plan that minimizes the total cost or the expected cost. Okay? Mm-hmm. And now modern versions of this involve a bunch of different bells and whistles, all of which are useful to us. So for example, you can regularize this objective function by adding a regularization term that promotes entropy in the transport map. And this is called entropic regularization. It was developed by, I believe, by Marco Chiuturi, although most recently, he developed it for algorithmic reasons, and it really speeds up the computations. But this line of thinking actually goes all the way back to Schrödinger, who was thinking about relativistic quantum electrodynamics from a statistical point of view. And he was imagining that you have n indistinguishable particles. And these indistinguishable particles are all kind of undergoing Brownian motion. And so you observe where they are maybe at one point in time, and then you might observe where they are at the next point in time. So just to review, if you just had a single particle that was undergoing Brownian motion, then the distribution of where it would be at time t is a normal or Gaussian random variable with variance that depends on t. And it depends on t and also the diffusion constant. Okay, so if the diffusion constant is bigger, it'll be a wider Gaussian. Actually, Schrödinger was considering now the problem where you have a bunch of different particles. Okay, so if you have n indistinguishable particles, you're not really sure which one matches to which one at the next point in time. And so you also have to kind of identify the matching or coupling of these particles. And if you assume that they're undergoing Brownian motion with diffusion parameter epsilon, then the maximum likelihood coupling of the particles at time 0 to the particles at time 1 is given by an entropically regularized optimal transport problem.
Yeah, this gets confusing because, of course, there is a different but, I guess, related notion of coupling in quantum mechanics, right?
I'm not familiar with that.
Or I guess they call it entanglement, right? Entanglement. Particles.
Yeah, sure. So there's no quantum mechanics here. Just to classically assume that they are undergoing Brownian motion.
Okay.
But you just don't know which particle is which. So, if you observed, say, two particles, and one was red and one was blue, and you observed them both at time 0, and then you also observe them at time 1, then you would know that the blue one went to the blue one and the red one went to the red one. And since you wouldn't really know exactly where they were in between, there'd be some distribution over possible paths. And in probability theory, that's called a Brownian bridge. But now, if the particles, instead of being colored red and blue, are all black, then you don't know which black particle at time 0 gave rise to which black particle at time 1. The distribution on all possible paths to go from this set of indistinguishable particles to another set of indistinguishable particles is called a Schrödinger bridge, like the extension of a Brownian bridge.
And I guess the amount of regularization here depends on the time, right? The longer you wait, the more you have to rely on entropy and less on the observation themselves.
That's exactly right. Or if the particles are diffusing faster, there would be more entropy in kind of which particle went where.
Okay. And so you use this to determine which cells went where. But I guess we came here from the discussion of cell growth, right?
Exactly. So there's a paper by from the Konrad Hochtinger's lab on reprogramming, viewing it as kind of a two-state process. Okay, like a continuous time Markov process, but with two states.
Before that, do you want to introduce this notion of reprogramming if someone's not familiar with it?
Oh, of course, yeah. One particular developmental process that's kind of, easy to work with and, quite reproducible, but still there's a lot of interesting questions, is this notion of cellular reprogramming. And this is the data set that we are actually working with in our paper. So in 2007, Yamanaka introduced these four factors that you can use to kick a cell out of its comfort zone and reprogram the cell from one stable equilibrium in gene expression space into another stable equilibrium in gene expression space. So you can change the type of a cell by giving it a virus that induces the expression of some genes. Now, in our data, we were using secondary mouse embryonic fibroblasts. So we actually had kind of a genetically pure mouse that already had all of these viruses already in the genome, and we could turn on these genes with a small molecule named doxycycline. So if we give the cells doxycycline, then they start reprogramming, and then it takes about 2 weeks for them to reprogram. But not all of the cells kind of go in the same direction. Not all of them successfully reprogram. Some of them die, and some of them turn into neurons, and some of them look like they turn into placental cells. So there's kind of some branching process going on. And at any point in time, a cell's fate might be partially or fully specified. And so this kind of branching phenomenon, we're trying to capture these cell fates with the transport maps. Okay? And I was about to explain cell growth and how we include that in the transport maps, and also how we specify our growth rates. Okay, so let me first explain how we include growth in the optimization problem. Okay, so what we have a convex optimization problem, and the optimization variable is this coupling matrix, okay? So if we actually have a finite number of samples from these distributions, we can set up this finite dimensional optimization problem, right? In general, a coupling is this infinite dimensional measure that could couple one distribution to another distribution. But given a finite number of samples, say n samples at time point 1 and m samples at time point 2, then we can form the empirical probability distributions at these 2 time points, and then solve for the optimal empirical coupling between these 2 empirical distributions. So that's actually, just growth aside, that's how you can set up an optimal transport problem between 2 empirical probability distributions. Now, in the presence of growth, optimal transport might do something funny. And to see why, here's kind of one picture that you could think about. Imagine that you have a mixture of 2 subpopulations of cells. And at one point in time, say they exist kind of in equal numbers, okay? And then at the next point in time, suppose subpopulation 1 is growing kind of more rapidly in the intervening time, and so that when you sample from this distribution at the next point in time, you obtain an unequal mixture. Say population 1 has doubled in size, then many more cells will be in subpopulation 1 at the next point in time, and if optimal transport didn't know about this, it has conservation of mass fundamentally built into it. So it would actually steal some cells from population, the second subpopulation, and teleport those over, instead of kind of allowing subpopulation A to give rise to subpopulation A, and also grow, and subpopulation B to give rise to subpopulation B and also move and grow. And so this notion of growth is something that we included in our optimization framework by modifying the constraints of the optimization problem. So the constraints of the optimization problem classically are just that this coupling has to be a doubly stochastic matrix, which just means that the coupling is a joint distribution that has kind of the prescribed marginals.
Yeah, so the sum within rows and columns are constant or equal to 1.
Should be 1. Yeah, exactly. Should be 1. And people use optimal transport in this context to match one group to another group. But now, if the objects themselves are allowed to grow, so you might want to match one object to, say, 2 objects at the next time point if it's doubling in size. So yeah, we just modify the constraints in exactly that way, right? So we just say that each cell is going to give rise, on average, to a certain number of cells at the next time point, and then we take that into account when we're matching the cells. And so basically we modify the amount of mass that's coming out of each cell, and that just amounts to rescaling the empirical distribution at the first time point, so that there's non-uniform weights on all of the cells.
Right. But the growth function is given by you. It's fixed,
Yes.
right? As opposed to making part of the optimization problem itself, like trying to estimate. Because in the example you gave, where we have this mixture of two cell populations, of course, the optimal transport might assign or transfer some of the cells from population 1 or subpopulation 1 to subpopulation 2, but there could also be some cost associated with this. And
Right.
I imagine it would be possible to set up this optimization problem. I have no idea how tractable it is or how much data one needs to estimate everything.
Yeah, actually, in theory, I don't think it's identifiable because just from population measurements. So if you just have one distribution, if you imagine that there's actually a stochastic process, okay? I like to call this actually a birth-death transport process because it's like the classical concept of a birth-death process describes the number of individuals in a population over time, right? And like a pure birth process, for example, is one example of that is like a Poisson process, where you have births arriving at some rate. If there's in some infinitesimal amount of time, delta t, the probability of a birth is specified at some parameter beta, and a death is specified by some parameter delta. Then the expected number of individuals over time is e to the beta minus delta times time. That's a classical notion of a birth-death process, but we're kind of working with this concept of a birth-death transport process, okay? So this birth-death transport process allows cells to move and grow and also transport. Cells from a subpopulation can give rise to different numbers of cells, and I think estimating the growth rates is certainly kind of a fundamental will have a fundamental impact on kind of the quality. But we have this knob that we can tune in the algorithm, which actually specifies kind of how confident we are in our estimate of the growth rates. And that's where the unbalanced transport comes into play. And that was developed by Linek Shiza, who I mentioned earlier. He's a fantastic graduate student, now postdoc in France. He was working with Francis Bach for his postdoc. So yeah, we do have to specify the growth rates in advance. And I think they could be measured, easily with some additional data, and identifying the growth rates of subpopulations is kind of a smaller problem in some sense than the entire problem of figuring out which cell goes where.
Okay, I see what you're saying. I'll make a final attempt to pitch this idea to you.
Sure.
So I guess what I'm saying is that if you measure this process on a very fine time scale, then you can tell. So, for example, you have your two subpopulations that are sufficiently far enough, right? And if you assume that your time step is small enough, then you can be reasonably sure that these two clusters, they remain apart. And so whatever quantitative changes are within those clusters, so one cluster got bigger, the other one got smaller, that corresponds to your growth or death.
Yes, and I agree. In that setting, I completely agree.
And since you didn't take this route, I guess that your time step is not fine enough to make these assumptions?
We used stuff like this to give kind of a rough order of magnitude and just inform our estimates of the growth rates. So your intuition is completely right. If you have well-separated subpopulations, then the transport problem then is kind of trivial in some sense. You basically have two basically decoupled processes that are proceeding in parallel. So this is kind of a very easy version of the problem. But this wouldn't work well in kind of the in and around branch points where the process is doing some very complicated stuff. Yeah. So in general, our method does rely on having a rough estimate of how much cells with different signatures will proliferate in the intervening time intervals.
Okay. Another related question that I wanted to ask is, so you sort of juggle between discrete entities like discrete distributions and discrete cells and continuous stuff. So one instance is that your growth rate is continuous, right? But then your distribution is just a set of discrete points. How do you reconcile those two things?
Yeah, there's actually a very nice, elegant theory that I'm kind of building up in the meantime since posting the empirical work. And this theory is for these birth-death transport processes, okay? So like imagine that you have a stochastic process indexed by some continuous time, and also it's a distribution on, it could be a continuous distribution on gene expression space, okay? Mm-hmm. Now this is kind of some population level quantity that if we had infinite data, maybe we would have access to. And suppose it has some true coupling between time points, okay? So let's say we call the true coupling from time t to time s gamma of ts. So gamma of ts would describe the full true joint distribution between a random cell at time t and a random cell at time s. Now, another population level quantity is the optimal transport coupling between times t and time s. And this is another quantity that we can't actually compute because we only have a finite amount of data. But this is the coupling on the true distribution at time t to the true distribution at time s that minimizes the cost. And there could be entropy regularization and other fancy stuff like that too. But this is defined in terms of the solution to an infinite dimensional optimization problem. Now, given empirical distributions, it's well known that, for example, the mean of an empirical distribution will converge to the mean of the population, right? Like the law of large numbers specifies this rate of convergence, and the central limit theorem specifies kind of the fluctuations that you would see in any finite sample.
Yeah, if you increase the sample size.
Exactly. So as you increase the sample size, the fluctuations will, converge at 1 on square root of n or something like that. And this is like a parametric rate, because here the mean is a parametric quantity. And it's actually kind of well understood that transport maps that you define on empirical distributions will asymptotically converge to this population level optimal transport map. So the empirical transport maps that we compute from our discrete, finite data sets will in some sense converge to this true not the true coupling, but the optimal transport coupling given infinite data. And the magnitude of their fluctuations and their theoretical properties is something I'm working out now in collaboration with Philippe Rigolet and Jonathan Weed and Linek Shiza. Now it's a separate question as to, now here's kind of the key modeling assumption, is that for this stochastic process, in order to try to estimate the coupling, we're making the modeling assumption that the true coupling, this gamma, is close to this optimal transport coupling.
Yeah.
And that's kind of the key modeling assumption. And but I actually think it's a fairly natural modeling assumption because it basically assumes away all of the stuff that would not be identifiable. And by that I mean these kind of stationary dynamics. So if you want, I can give you a better idea for the types of dynamics that may or may not be identifiable from population level, like just sampling from a distribution at different points in time.
Before that, so I'm trying to understand. So I understand that given the stochastic process, the true coupling is, of course, a well-defined entity, but the optimal transport, doesn't it depend on what your time step is? Because if the time step is long enough, then it's hard to draw any connection because everything sort of moved around.
Yeah, you're exactly right. Yeah, so I meant to say that on very short time scales, the optimal transport coupling might agree with the true coupling. So you can think of this stochastic process just as kind of a point describing a curve in the plane. And the optimal transport coupling is in some sense a geodesic or a straight line in distribution space. And so the optimal transport coupling, so if we assume that a stochastic process is doing optimal transport on short time scales, it means that this curve that it's making can be approximated by, on very short length scales here, by little piecewise linear line segment, by some, yeah, and as you point out, on large timescales, you're exactly right that this is not reasonable.
Okay. Yeah, so you were talking about the parts that are not identifiable.
Right. So now imagine that you have some stationary dynamics. So what do I mean by stationary dynamics? I mean that suppose you have a distribution that's actually not changing over time, but the individual particles, the individual cells are changing over time. For example, suppose the cells are all doing the cell cycle. Or hematopoiesis is another kind of classical example of the entire system is more or less in equilibrium, but the individual cells are going from some stem cell state to some eventually fully differentiated state, and then maybe dying along the way. So if you just think about a distribution on the plane, just to get a very concrete mathematical example, suppose we just have a distribution on the circle, and the cells are spiraling around in a circular pattern, but at all points in time, you might just have the uniform distribution on the circle. So you wouldn't be able to tell just from, even if you got infinite data, right? The samples, at each point in time, you would get samples from this uniform distribution on the circle. But if they were independent samples, even with infinite data, you would see very well that you definitely have the distribution on the circle, but you wouldn't be able to estimate the coupling at all. And actually, optimal transport would make kind of a simplifying assumption that this coupling is actually totally stationary. So the points are actually not going anywhere. So that's what the optimal transport coupling would give you in that specific example. And it wouldn't agree with the truth, even if you looked at very short timescales.
Right, but that assuming that it's perfectly uniform, because if you have any noise, you will be able to observe that the noise moves around the circle as well.
Exactly. Yeah, if you had just one slice of the pie, that pie slice would kind of circle around. But those would be non-stationary dynamics then, right? The distribution would be changing over time.
I guess. So I'm thinking if your distribution is stationary, right, but then you're working with the samples from the distribution, samples are noisy, and wouldn't you be able to observe that noise in the samples while the underlying distribution remains stationary?
So let's see. So in the setting where you're sampling from the entire uniform distribution on the circle, I agree that at each point in time, you would have a different empirical distribution. But as you get more and more data, that empirical distribution would converge to the uniform distribution. And so it would actually be stationary in that sense. And so I don't think you could just leverage the noise to learn the dynamics in that simple example, at least.
Yeah, maybe not.
Yeah, yeah, maybe not.
Right. So we were talking about the connection between the continuous stuff and the discrete stuff, and you're saying that the discrete optimal transport that we get from the empirical distribution should converge to the idealized optimal transport, and then the question becomes whether the idealized optimal transport is close enough to the underlying stochastic process. But in the practical implementation, how do you reconcile the continuous growth with the discrete distribution? So, for example, if your growth function tells you that the cell at this point in space grew by 20%.
Yeah, good point. So the way that we do this is we specify, some kind of prior distribution on how much we expect a cell to grow. And that's in terms of the birth and death rate. And then we just kind of subtract those and exponentiate it. And that gives kind of the expected numbers of descendants that cell should have. And then the algorithm tries to match that as well as possible, but it doesn't, it's not required to match it exactly. And also because the transport has some entropy to it, so if a cell is really going to transition to one specific point, if it's not going to divide, it'll give rise to one cell at the next time point if it doesn't die. And if we're not exactly sure which of our cells is the best representative, if we're not sure which of our cells is the best kind of putative descendant of that cell, then the algorithm will spread some probability mass over some, kind of cells that we see at the next time point. So there's entropy in the transitions of a single cell for biological reasons, in the sense that, a single cell may not have a totally specified fate, and then it really makes sense to have kind of 50% of its mass, in one place and 50% of its mass in another place. But then also just for purely technical reasons, we might, put some transport mass on a little cluster of cells hopefully surrounding where the true descendant might have been if we'd measured it, Just to specifically answer your question about the continuous growth rates, though, right? Like, if we say a cell is going to give rise to 1.72 cells at the next time point, what does that mean? Is that your question?
Yeah, I guess it makes more sense if you think about like very dense clusters, when you can expect that if your growth rate is 1.72, then that percentage of cells will divide, right? It makes less sense if you consider an isolated point in the gene expression space where
there may not be 1.72 cells to map to it.
Yeah.
Yeah.
But again, we're just sampling, right? So in the actual biological process, there are probably enough cells.
Right. Yeah. And this is, so 1.72 is kind of the expected growth rate, right? And and that means that maybe it gives rise to 2, and that's a little bit more likely than it giving rise to 1 descendant, but it could also die, or give rise to 3 descendants. But we kind of think of that as the expected number of descendants. And so that's how much mass we hand a cell. So we say, okay, you specific cell are responsible for allocating 1.72 mass of descendants at the next time point. And then we take this distribution at the next time point and we say, okay, we saw, 5,371 cells at this next time point. And then we say, okay, given that we had this many cells at the first time point, we scale that number up by the overall average growth rate of all the cells at the first time point. Okay? So if there's an overall growth rate of 2, then we'll have roughly 10,000 total. Sorry, I actually said that a little bit incorrectly, right? So we take the number of cells at the first time point and we scale it up by the average growth rate, and that determines kind of the total number of cells, the total number of seats we have to fill at the next time point. Okay.
So I'm thinking about this degenerate example, and maybe I'm doing it wrong by concentrating on the degenerate example. Okay. But if we have this isolated point, you get only one sample from this patch in the gene expression space, and it is just 1 point at time 0 and just 1 point at time 1. But the equation tells you that it produced 0.72 offspring. Will you move this mass elsewhere, or will you just say that, okay, the expected growth is this, so I'll just assign greater mass to that isolated point?
Yeah, so we would probably throw the extra mass away because we're doing unbalanced transport. So unbalanced transport, it actually takes the constraints of the optimization problem. Now remember, we modified the constraints to include growth, but in classical transport, these are equality constraints, like you're looking for a doubly stochastic matrix whose rows sum precisely to 1. And then now we just modified that aspect of it so that the rows don't have to all sum to the same number, but they should all sum to something. But then the final layer of it is that we relax these equality constraints to KL divergence terms in the objective function. So the overall objective becomes we want to minimize the total transport cost, plus we want to promote the fact that each cell has roughly as many descendants as we think it should. And precisely in terms of this KL term, in Kullback-Leibler divergence term in the objective function.
Interesting. So yeah, I thought when you said that this is an unbalanced transport problem, I thought by that you meant that you have this growth factor, but you actually, there's an additional imbalance in that you don't even try to match that exactly.
That's right. Yeah. And so that's right. So Laennec-Shizah invented unbalanced transport in the kind of the abstract sense. And then we applied it here, with our inclusion of growth rates to just say that we don't have to know the growth rates exactly. And each cell doesn't have to give rise to exactly as many descendants as we say it should. So it's okay if it throws away a little mass here or there, or, gains a little extra mass here or there.
Yeah, that makes sense. Okay, I think we wandered enough in the mathematical weeds. So
okay.
let's talk a bit about the biological applications and implications of this method. Maybe we'll start by what your input data looks like. So we mentioned that you get single cell gene expression data at some time slices. So what is your actual time step that you use in your experiments?
Good. Yeah. So this reprogramming process starts out with mouse embryonic fibroblasts at day 0. And, we have 10 time points over the course of 16 days. And we have 65,000 total cells with between, 5 and 7,000 data points at each time point, with the exception of one of the time points that didn't have any it only had one biological replicate. So that time point actually only has about 1,100 cells at that day 11. So yeah, in general, we have between 5 and 7,000 cells at each time point, spaced 1 or 2 days apart.
Okay. And then you apply this algorithm that infers the transport map. So how do you visualize or how do you interpret that data then?
Right. So given our transport map, what we can do is we can look at the descendants or ancestors of any individual cell or any population of cells. So basically there's a row of this transport map for each cell at one time point, and a column for each cell at the next time point. And so what a specific row tells you is the distribution of descendants. So the total sum of the row tells you the number of descendants that cell was actually allocated by the algorithm, and we rerun this several times with different, regularization parameters until we see that, our constraints are kind of 90% correlated with the growth rates that we input. So the input and output growth rates are kind of correlated to some extent. Yeah, and then we interpret these transport maps as telling us, given a cell at one point in time, you can compute its descendants immediately at the next point in time, and then you can also take those descendants and push them again through the transport map further and further into the future. Or you can look at the ancestors all the way back into the past over multiple time steps by just composing the transport maps from the appropriate time intervals.
Right. So you can say, for a given cell type, you can say what its possible fates are and how likely it is to go to those fates.
Right.
And how do you present this in a way that's comprehensible to mere humans?
Yeah, so the sequence of transport maps is a little bit hard to digest. What we do is one thing we do is we visualize the landscape of the cells. So my wonderful collaborator, Marcin Tabka, has this beautiful technique to visualize the cells using the force layout embedding. And this is kind of one of the key figures in our paper. And basically every figure in the paper uses this visualization of all of the cells laid out as a two-dimensional landscape, right? So that's how we just visualize That's how we visualize all of the data, right? And then you can annotate this landscape with signatures. So, which cells are expressing any individual gene, which is maybe corrupted by dropout, so maybe you only get a few cells expressing, or which collection of cells are expressing, some collection of genes in a gene signature. Or, given any group of cells, what does the descendant distribution, which is, a finite discrete distribution on the cells from the next time point, what does that probability distribution look like? So which regions of gene expression are you more or less likely to go to? We can also cluster all of the cells and reduce these large transport maps to summaries that say, what's the total mass coming from one cluster at this time point and going to another cluster at the next time point. And, we can see, like, in terms of these clusters, so maybe there's one cluster of cells that's very relevant, so the successfully reprogrammed cells, so which cells are more likely to give rise to those, So we have a bunch of different ways to analyze these transport maps that we outline in our paper.
Yeah, so you mentioned that this helps you derive regulatory models and gene modules.
Yeah, so that's an interesting way that's one way that we can also try to summarize the transport probabilities. And the most general framework is, this idea of learning a regulatory model. But kind of the simplest way to think about it is just like consider iPS cells as a destination, induced pluripotent stem cells, and ask which cells at the previous time point are most likely to go there, and which cells at the previous time point are least likely to go there. And then, given those two classes, what are some genes that are helpful in discriminating between those two classes? And then we, maybe those genes are involved somehow in the reprogramming process. Now, these regulatory modules are kind of one step more sophisticated than that. So for the regulatory models, we assume that there is some kind of differential equation, and that's governing the growth and the transport. It's actually governing the transport. So suppose if a cell has some gene expression profile X at time t, then maybe it has some rate of change x dot. So dx dt is f of x.
Where x is this whole expression vector.
Exactly, yeah. So x is a 20,000 dimensional vector, and we're trying to estimate the function that defines some differential equation that specifies how the cells evolve over time. And obviously, this is a very high-dimensional problem, so we have regularization and stuff, but how do you actually set up some regression to learn this function f? So a priori, it seems quite hard because we don't have paired samples, right? We don't have any coupled samples. The generic thing in machine learning or statistics, to solve a regression problem, you generally have like xi comma yi pairs, and you want to say y equals f of x, maybe plus noise. So in our framework, we say, like, first we solve the transport problem, and then given a cell x at one point in time, we can sample likely descendants of that cell and then set up this regression framework to try to learn this function f from these data that we generate from our transport maps. And this f is this regulatory model that we learn. And we have these regulatory modules. These modules just come from the specific functional form that we give for x as a low rank linear function.
Interesting. Yeah, that's very cool. That's very cool. I guess the other thing that people do in this space, which is probably much simpler and more limited, but we often hear about pseudotime. So how does this compare to pseudotime analysis?
I wouldn't say pseudotime is simpler or more limited. So pseudotime analysis, this is at the very core of all the issues. So pseudotime analysis is the version of this problem where you don't have these explicit temporal labels.
Yeah.
Right? So I talked earlier about stationary and non-stationary processes, right? So the pseudotime approaches are entirely designed for the potentially stationary setting, Whereas our approach couldn't hope to work for the stationary setting, like I described earlier. And actually, even more than that, it essentially assumes that all of the stationary stuff is basically zero. So, I mean, put differently, it just returns to you the purely non-stationary components of the dynamics that are driving the process. So the pseudotime methods might hope to learn even the dynamics leading to stationary, a stationary sequence of distributions where the distributions are the same at all. If you only have one time point, then your distribution is just kind of stationary. Like in hematopoiesis, for example, you don't have labeled time points. So I see this as a kind of a purely complementary approach to the suite of pseudotime methods, where we're entirely going after these purely non-stationary components of the dynamics.
Yeah, I guess what I meant was if you, as you say, if you have just one time point, or if you have measurements at very long time scales, or if you have maybe unrelated measurements in like unrelated organisms, then you don't have this temporal correlation, and you can sort of assume that this is a stationary distribution, but it gives you much less data to work with. Whereas if you sample at high frequency, then you get much more data, and so you presumably can make stronger or more interesting conclusions from that. Would that be fair to say?
Yeah, I mean, it's fair to say, but I mean, if you have more data, that's always better though, right?
Yeah. And so what's the current status of the software? Is it released? Like if someone wants to try it out, can they do that? Yeah,
so we have a version kind of 0.1 of the software package, and it's available on GitHub. We haven't kind of made some giant public announcement of it yet, and that's because it's still at the stage where it needs a little bit of help. But if people are interested in taking a look, it's at the stage where you kind of use at your own peril, but also with help from us. So it's it's under rapid development though. So yeah, it's on GitHub as WOT, like for Waddington Optimal Transport, on the Broad Institute GitHub website.
What's it written in?
It's in Python.
Mm-hmm. And so to finish our discussion of the biological application, is it simply an exploratory tool, or did it actually lead you to any conclusions, any new biological knowledge, so to say?
Yeah, so the main biological knowledge that came out of this, one of the main kind of pieces of kind of pseudo validation for the method also came from the regulatory modules. So we looked for cells that were kind of more likely to transition to iPS cells, and we also fit this regulatory function F, and we saw some list of genes that looked kind of very important. So kind of the top genes that were most likely to drive you in that direction of successful reprogramming. And then we experimentally checked this list. This part was done by Jian Xu, who's another one of my wonderful collaborators, and he was kind of one of the central driving forces of this project, from its inception. So Jian experimentally validated this gene OBOX6. We saw that it increased the probability dramatically that cells would actually successfully reprogram. So yeah, and the analysis also brought this up as one of the top genes that were most important in driving you towards successful reprogramming. Yeah, so in that sense, it is kind of a tool for exploratory data analysis, right? Just kind of by definition. But it's also kind of useful and can help shed light on various processes like reprogramming. But it's also, we hope that it's generally useful. And a bunch of people in the Broad are starting to use it for a number of different things. So that's exciting. Cool.
I also wanted to ask you, like, how did you first find out about optimal transfer? Like, was it a part of your toolbox already? Or I think you mentioned that Eric also contributed to this, right? Was it his idea?
Yeah. No, it was my idea to specifically consider optimal transport, but it came out of a conversation with Eric. So I was aware of optimal transport just generally from my training in theoretical statistics. Mm-hmm. And also I'd learned about it recently through conversations with Philippe, just kind of abstractly. He had a kind of a group reading group on optimal transport. So optimal transport was on my mind, just for kind of purely theoretical reasons. And then out of conversations with Eric, we were thinking about, if you had a school of fish and they were swimming, and then you didn't know which fish was which, so how would you see how to connect them to their versions at the next time point? And at one point, Eric had an idea, maybe we can do something with some doubly stochastic matrix. And so he called me on the phone to announce this idea. And at that point, I immediately said, this sounds a lot like optimal transport. Maybe something like that could be useful. And then so I started reading more about optimal transport and some of the recently developed algorithms to do it. And at first it wasn't working very well because we didn't realize that we had to use these growth rates. So I learned just from doing some examples with mixtures of Gaussians moving around in 2 dimensions that these growth rates were going to become pretty important. And then during the time, about around the same time when I was doing these simulations with mixtures of Gaussians, Laennec, Shizat put his paper on unbalanced transport online as well. And then so I realized we could use that to actually solve this numerically very quickly in an unbalanced transport framework, as opposed to just kind of hard constraints framework like ordinary optimal transport.
Very interesting. Do you think this will become like the new hot thing? Like we had neural networks that like everyone ignored for a long time, and now they're the new hotness. So do you think there will be an increased interest?
Yeah, I mean, certainly in the statistics community, like it is kind of a fairly hot topic. Optimal transport is, there's a workshop entirely devoted to optimal transport in Overwolfach, which is like a mathematical research conference setting. And so in the theoretical statistics community, it's certainly picking up. And, I kind of see this as one of the first major application of optimal transport in biology, and also to time series data. So I think this framework of estimating the couplings of stochastic processes from independent measurements could have applications, in diverse scientific settings, like anytime you have a stochastic process. And I've really enjoyed working on this project because, coming from a theoretical statistics background, I don't have to leave behind the math entirely, but also it seems to have, be starting to have like big impacts also in single cell RNA sequencing, especially in the time series setting, which I think is also an important direction for single cell analysis in the future, is more kind of temporal analysis, because, it is very natural to consider temporal processes, especially in developmental biology. It's all centered around the concept of a developmental temporal progression. So I hope that this will be, a big thing in the years to come.
Awesome. All right. We've been chatting for a while. Is there anything else you'd like to mention before we wrap up?
No, this is a great conversation. I really enjoyed, talking with you.
Yeah, Geoffrey, I enjoyed it too, and I hope our listeners will as well. Thanks a lot for coming on the podcast.